Our next speaker, Indrajit Ghosh. Dr. Ghosh is Associate Scientific Director of Biologic Product Development at BMS. And one of the topics that we have been discussing this morning was about subcutaneous simulation and analytical for it. But here we are talking about high drug load, which is always a limitation for injectables. So I would like to learn more about how you have addressed the antibody dosing at high drug loads. Thank you.
Hello, everyone. And thank you, Simon, for giving the opportunity to come here. I know we are working for a long time. Previously, I have given the talk on the milk extrusions and nano, and the last 5 years I moved to biologic drug product development. Is a great learning from me, and today I'm going to present on the roadmap for development for subcutaneous form for the high dose. I'm emphasizing the word high dose. It's not just a high concentration, it's also the high volume, and some of the challenges with the biopharmaceutics. Is the audio, it's fine? Okay. Thank you. So I'm going to cover today about some of the challenges with the high-dose monoclonal antibodies, the strategies, the development strategies, how to design the drug product, and taking into consideration the two attributes, the high concentration and high volume, especially the viscosities, some of the novel drug delivery technologies, the device options. And in the second part, I will be discussing about the biopharmaceutics, some of the key attributes from the drug substance, the formulation aspect, and also the delivery technologies and device. So 50% or more of these proteins are now getting high dose. It's more than 200 milligram, but again, these numbers keep on growing. Now it's going to 1 gram or 2 grams. In subcutaneous, so that's the biggest challenge. And obviously, it can be given by the IV infusion, which is still one of the common approach in all the disease areas. But as you know, the IV infusion has some complexity with the patients, has to go to the hospitals or clinic, is a high cost. So by 2030, it was speculated that standard of cure will be subcutaneous administration, even in the oncology area. And because ADVI is getting popular, so as I just discussed, it's a self-administration at home, easy of use, and obviously the cost benefit from insurance. It's also more patient-friendly and also considered from the healthcare practitioners. But the challenges are generally subcutaneous administrations. The max volume is 2 ml, so 1 to 2 ml. So that was being used so far. But now, as we are getting more high-dose drug, let's see how we'll take care of the challenges. We recently actually published one paper. So myself, Ramesh, is sitting here. And some of our colleagues from BMS. So there are many antibodies in the market. So as you see in the graph, in the last few years, in the last 20 years, about 120 antibodies, and there are more. So this graph is until, I think, 22, but there are a few more approved recently. But so among the 120 antibodies, there are about 45 high concentration antibodies. So we have done some of the insights from the product, including formulation aspects, the delivery technologies, what kind of devices they have used, and other challenges. So in the next few slides, I'll be referring this paper and also talk about the other challenges. So again, this is a snapshot from all the disease area. As you see, some of the drugs from antibodies, fusion proteins, and small molecules. The green one is antibodies, and the orange is the fusion proteins. There are some blockbuster molecules already there in the different disease areas. So obviously, in the immunology space, there are more high concentration antibodies or the high dose antibodies which makes sense because this is more like a chronic disease area, and so this is getting more popular. In the right-hand side, I have shown the different concentration of the antibodies. So in our research, we have taken like 100 milligram ml or more. So as you see here from the statistics, like 100 to 150 milligram, there are more antibodies, but still it's growing. About 150 to like 200, there are also a few more. From the route of administration, there are a few IV in the high concentration, but most of them are subcutaneous. And this subcutaneous, if I talk about the biopharmaceutics part, it gets absorbed or it administered in the hypodermis area. So that's the complex structure. It's a highly variable thickness. And as you see here, we can deliver up to 2 ml without any problem. But as the increase in concentration, there are other challenges for the absorption. Because when we talk about the bioavailability, it is the absorption, so mostly it gets absorbed from the lymphatic channels. So we need to consider the absorption from the lymphatic channels, which is the major route of the absorption. Some remaining portion also get absorbed from the capillary, and also there are a lot of clearance factors up there. So all these things have to be considered. So when we actually switch from IV to SC, generally, so far the antibodies have shown about 50% to 80% irritative BA, and this is because of some of the challenges we saw here, some of the clearance, the precipitation, and all other factors. So this is one important area that we need to consider for the biologics. It's not as bad as the small molecules, because we have seen for the small molecules, it's 2%, 10% is there, like BCS class 4. But here, the impact is more. Even a 10% reduction of the bioavailability will impact the drug amount, right? And as you know, biologic drug substances are highly expensive. So any reduction of the bioavailability actually impacts the expense, the cost of goods of the drug product. And that's why it's so important to consider. So the problem statement. So I'm talking about 2 ml, but now we are getting the antibodies, the dose from 1 gram even to 5 gram in some of the disease area of unmet need, like Alzheimer's, because it has to cross the blood-brain barrier, right? So the dose is getting high. So 5 gram to administer in the sub-Q. So that's the problem statement. And how we tackle that? As I just discussed, in high concentration formulations, large volume device, and considering all the biopharmaceutics, we design the product and I try to understand the bioavailability, and also the other important part is the tolerance, the injection site reactions, the pain. So these are the things that we also need to consider for patient compliance. So let's first start with the high concentration formulation. So the challenges, solubility, the viscosity, and aggregation. You see in the graph is increase by geometrically. So until 100 or 150 milligram per ml in the bottom graph. So this is the protein concentration versus viscosity. So as the concentration increases, the viscosity also increases. And this has different implications in the process and also from the product standpoint. Like drug substance process, it has to go with the shear stress, interfacial stress during the UFDA process, the filtration process. There's a backflow. Same thing for the drug product. There are challenges in the process and also during the filling. Same thing for stability. So as the viscosity increases, this has an impact on product aggregations, the particulate formations. So these are the challenges. Device, so nowadays, like device can able to handle high viscosity, but also there is some limitation considering the force or injectability. And so these are the excipients that we generally use in this type of formulation. I'm not going to just go into the details, but these are like a buffer, which is mainly used to maintain the pH, because some of these excipients will be actually discussed in the next few slides as the key excipients or the key formulation that we need to consider for bioavailability and also for the tolerability. So buffer is one of the important excipients, the tonicity, to maintain the osmolarity, the surfactants, and then the viscosity reducing agent beside other ingredients like antioxidants or chelating agents, those are all anyway there. So first, let me start with the viscosity. So this is again from the high concentration antibodies that are commercialized. As you see here, the most commonly using excipients are the arginine, the glycerin, the sodium chloride, the lysine, the proline. So this is one of the study that we have done. We have used arginine, sodium chloride, lysine, and glycine, and this is one of the formulation approach that we start. We see the impact of some of the excipients on viscosity and also on the stability. So again, we have seen arginine generally work in many cases, but with few exceptions. But we still need to do some screening of these excipients. And here we see arginine and the sodium chloride work good because it tried to actually protect some of the protein-protein interactions and maintain the viscosity. We check the viscosity profile at different temperature, but 20 or 25 degrees centigrade is the most appropriate because where we do all the processing, and also this at the temperature where the patient generally take out the product from the refrigerator and thaw it. So this is the temperature generally we try to assess the viscosity, but the lysine and glycine, this is more. And the same thing we have done from the size exclusion chromatography to understand the high molecular weight, the particle formations. Again, arginine and sodium chloride work better than lysine and glycine. So before the formulation development, we try to do some kind of modeling. So in this slide, we are showing, so this is a modeling for screening some of this excipient by using the site identification, the ligand competitive saturation. So here we do, so this is again a prediction. This is not actual study. We do the mapping of these proteins to understand what are the sensitive area or what are the areas that will be more prone to the protein-protein interaction. So in this picture, in the 3D picture, you see the white portions are, it is less than 20% probability of interaction. The red having more than 50%. The orange is in between. And then we also do the mapping of the different viscosity excipients, like lysine, arginine, and proline has done in this case, is coded by the color. And based on the mapping, we try to understand, or at least, rank order some of the excipients that will be most beneficial or that will prevent this interaction in terms of viscosity and stability. But based on this mapping, we select some of the excipients and do the actual study. So again, change the gears. So subcutaneous delivery for the high dose and for the biologics. So you see in this graph, so there is the viscosity and there is a dose volume. So these are the important factors. So as the viscosity increase, so in this blue area, these are some of the most commonly used excipients or the formulation we use when the viscosity is around 10 CPS. But as we increase the volume, then we have more specialized delivery technologies we use. And here are some of the examples of the formulation technology. Again, these are not all the technologies, but I'm giving some of the examples here, like Chimera technology and the Xeris Electrify. So these are some of the technologies by which we increase the concentration of the drug in the formulation. And there is also VAMI research. I will talk for each of these technology in the next slide. And same thing for the device. We have the pre-filled syringe, we have the auto-injectors, and then we have the patch pumps and then the infusion pumps to increase the volume. So again, the blue and the green area that are shown in the slides are some of the research area we are working now. But more than that, in the white area, there's the technology we need the development because the dose is getting more and more higher. So this is the snapshots of some of the delivery technologies. I'm not going to the details, but in a very high level. So Chimera Life Science, they have a platform of more than 200 different excipients. So they do a mapping of the different excipients and try to understand which one is the best for a particular antibody. The Electrify and the Xeris, we can increase the concentration up to about 400 milligram or more by maintaining the syringeability, and each have a different technologies like Electrify is using the ultra high the concentration to form the microparticulates, and the Zarish technology is using the spray drying. So these are some of the technologies which have shown a good proof of concept, but I think there are more has to be done in the clinical area, in the commercial area. The primary research is again not a technology, but it is based on some proprietary excipients, which is the nicotinic acid and tryptophan in combination. And there are a lot of work, other studies has been done to show that the concentration can be increased up to 200 milligram or more. So let's now go to the primary containers and the device part for the volume. Again, from all the antibodies on the high concentration, we have seen there are the vials, the PFS, the prefilled syringe, and the auto-injectors. But now we are going to a different direction. We have more the patch pump and the infusion pump, so those are needed. So in this slide, I'm discussing about some of the device technologies that we need for the large volume, more than 2 ml or more than 5 ml. So these are some of the examples here. The self-dose, it's a prefilled in a glass syringe or the auto-injector that can deliver up to 5 ml by using the enzyme hyaluronidase technology. And then there is also the Smart D. So Smart D is again a patch pump. This is a patient-friendly. So basically, a patch pump we can just adhere to or attach to the abdomen, and we trigger the injection until certain time, about 5 to 10 minutes. It can also be used without the enzyme, and the drug is delivered. And I'm showing here 2 examples: the Skydazy and also from Repetha. So both have been used the patch pump. So this is a high volume up to about 10 ml, and the subcutaneous has been delivered over 9 minutes. So the time is very important. So this time factor need to be considered, the volume and the rate of infusion. So these 2 are some of the primary factors that we need to consider for the PK and more for the tolerance. For the bioavailability part, so as I discussed before, the subcutaneous are mostly absorbed from the lymphatic channel. Now there are 2 factors that we need to consider: the molecular transport and also the catabolism. So these 2 are the primary factors that impact the PK in this route. But during the roadmap of the development, there are a few things need to be considered. Although we get the bioavailability of 50% to 80% in most of the cases, but in this area, the most important part is the comparability. So as we start from the preclinical, from phase 1 to phase 3, during the development phase, as we change some of the attributes like drug substance or the drug product formulation or the device, we need to ensure the comparability by doing up the preclinical or the clinical studies. For example, here the drug substance change. The glycan profile of this biologic drug substance, the sialic acid, this can have an impact on the PK and also the immunogenicity. Same thing for formulation, because when we switch from IV to SC, or the change of the concentration from low to high, and even change the formulation, we need to understand what is the impact on the PK and what is the impact on the tolerance. Same thing for device, the rate of infusion or the volume. This has also have an impact on the PK. So at each and every stage of the clinical of the roadmap, we need to assess and ensure the risk is less, and this is obviously some requirement from regulatory. So let me start with some of the preclinical, and I know today we have a good discussion from Mark on some of the CSER study. I'm not going to go to the details, but yes, this is one of the primary study we start with. It's not a full predictor of the human, but at least to screen some of the formulations or to understand any tolerability. We do CSER study to rank order some of the formulations, to understand the risk, because when we go from low to high concentration, and also we have to understand if there is any precipitation, because if there is any precipitation, this can impact not only the PK, but also the injection site reactions. There are also animal studies we do, and I think there was some good discussion yesterday and today about the prediction of the animal models, and we all know for the human, the best predictor is a human. So in this area, we still do some of the preclinical models, try to assess some of the risk, like rat, cyno, or the mini pig. And some of the animals are again not the substrate of these antibodies, so we need to also understand that. But the rat model is again for the first turnover, is not expensive, is a quick study. We do some rank ordering the formulations, and also to understand the impact of some of the glycan profile of the drug substance. But from the rat, the skin is very thick, so this is not a good predictor for the PK or the tolerance. For the monkey, and more especially the mini pig, are more better model to understand some of this PK and mainly the tolerance. So basically, when you go from IV to SC, we do some of the studies to understand the risk. It's not really a regulatory requirement, but just more for internal understanding. But we sometimes have to do the local tolerance study. So that is something important we need to file. The subcutaneous, so this is a slide I'm covering for the halozyme technology. So as you know, this is one of the technology that has been widely used for many of the commercial products I have listed here. And the dose volume, as you see, has gone from 5 to 15 ml. All the protein concentration lies between 100 or 20 ml. It's not very high, but the advantage for the technology is, so as you see in the picture, there is an enzyme called hyaluronin in the structure. So this is the enzyme that actually, or this is the protein that actually prevent the distribution of the drug when it's administered. Because of the hyaluronin, the drug, when it's in the high concentration or high volume, does not get dispersed. So this enhanced technology is basically we are administering or co-administering this hyaluronidase enzyme, which disrupt the structure of the hyaluronin and create the intracellular space of the channel so that the antibodies can be dispersed. And it got more space for absorption. And the same thing to reduce the pain and improve the patient tolerability. So we see some of the example, and also this enzyme, the systemic half-life is less than 5 minutes, so it's obviously biodegradable. This is a great innovation. And although we have seen some of the bioavailability of the rat has been increased in general from some of the clinical studies based on the published literature, there is a 20% increase of the bioavailability, which is also like a huge impact in this area. But again, it is not directly enhancing the bioavailability. What is actually doing it by disrupting the enzyme, it creates the space, it creates the channel so that the drug can be distributed and get absorbed more efficiently. So that's basically the benefit of this technology. The other part, the most important part, also the tolerance, the injection site reactions and other complications, adverse effect that can happen from these biological antibodies. So beside, again, the formulation, as we discussed, the PK, I think tolerance is also an important factor. So because when we talk about the tolerance, we have the formulations, we have the device, we have the patient factors. The formulation factors is the compositions, the buffer, the pH, the surfactants that we use, the viscosity. So these all impacts the tolerance. Same thing for device, the type of the device we're using, the injection rate and the volume, and also the patient factors or the human factors, which is also important. So this is one of the study we generally do because when you introduce a device, the PK compatibility studies, and also human factor study. In the left-hand side, we showed the impact of different kinds of device, like a pre-filled syringe or the auto-injector. We obviously do the impact on the PK. In the right-hand side, we use different techniques, but one of this is showing here the VAS, the visual analog scale, which is the pain factor in the patients, by using the different kinds of devices and how it's impacting the VAS factor, and based on that we know which is less painful. So this is again for the optimization of the study, and in the right hand side bottom curve, the green and the blue. So basically the green one is without the technology, without the halozyme technology, and the blue one is with the technology. So obviously the pain it has been reduced. So again, this is a heat curve. So the syringe injection force is also important, which is again directly proportional to the viscosity. If the viscosity goes high, this impacts the syringeability. and the force that needed to administer. Generally, this is, again, thumb rule, generally the upper limit of the injection force around 10 to 15 Newton, but this is just an example here. For example, we generally use the needle size from 27 to 30 gauge, but if the viscosity has increased, we generally try to keep the viscosity lower than 20 CPS, ideally less than 10 CPS for the high volume. If the viscosity is like 20 to 30 CPS, and we're using the 30 gauge needle, then as you see here, the injection force can go up to 80 or 100 Newton, which is obviously is going to be painful. So in the graph here, there's a heat map between injection volume and injection time. The injection time is also very important. We can increase the volume, but it has to be like infused in a slow infusion rate. So the green is less painful, the red area is having more painful to the patient. So obviously, in summary, patient tolerance, patient preference study we also do, because before we select the device, and also the tolerance, the injection site reactions. So these are all some of the parameters we need to consider before we select the device. The device selection is, again, very expensive. The development is very expensive. So we need to address some of these important considerations before we have the final device, like in the clinic. So again, going back to the formulation, so buffer and the formulation pH, and how it is impacting your some of the tolerance. So we use some of this buffer. Again, this graph from all the 45 antibodies that are of the high concentration that are being commercialized. As you see here, each has used like the histidines as buffer. Some of them also used the acetate, citrate. But these buffers are generally used to maintain the pH, because the pH profile is very important, as we all know, for antibodies, because it impacts the stability of the antibodies. Sometimes low pH can create isomerations, the high pH deamidation. So we have to have a very good understanding, optimize the pH range. Because if the pH is close to the physiological pH, then it is more tolerable to the patients. In high pH or the low pH sometimes can cause a necrosis and more painful. This is one of the a great example of one of the product, Humira, which is a blockbuster product. So Humira is first, when it's launched, with it contained the citrate formulation. As you see here from the pain curve, the pain factor, the VS factor is very high with the citrate, because citrate is a strong ion, so probably it has an impact for the significant pH shift. So this citrate formulation of the Humira is more painful. Later, AbbVie has launched the citrate-free formulation, which is less painful. So in this formulation, there is no buffer, and which is very surprising, am I right? But we know these antibodies itself act as a self-buffering agent. So basically, when you are increasing the concentration of the buffer, it is also helping to stabilize the molecule. So we had to have a good balance for selection of the buffer that are less painful, and also the amount that we use. So again, this is a great example for a buffer-free formulation by Humira. There are also other components that we need to actually address, like surfactants and tonicity agents, because surfactants that we generally use, the polysorbate 20, polysorbate 80, there are also few with polysomer, but surfactants can also cause the hypersensitive reactions. So we need to do some optimization by using the surfactants that will stabilize the antibody, at the same time it should be less painful. Same thing for the tonicity agent, we use to optimize the osmolarity. Generally, we try to keep the osmolarity around 300, which is close to the physiological. It can go up to 600, but again, I think there are more research need to be done with this high volume, because with the high volume, it takes time to equilibrate. So in general, in order to avoid risk, it's better to be close to 300. So this is the last slide for the roadmap for the development, so I'm giving a summary. So this is again a case study in which we have the dose until 1.8 grams. So when we start the preclinical studies, obviously, as I discussed before, we select some of the rat models or the mini pig models. So that time we had the early clone selections. So those who were in the biologic space, So in the early study, the clone is not finalized. So this is like an early clone or a pooled clone that we use for this, some of the tox studies or the tolerance studies in the animal. But as we go to the FIH, in general, we do the FIH with the IV because it has more flexibility with the dose ranging, as you see here, from 10 milligram to 1800 milligram based on the patient body weight. So by that time, we had the final clone selection. We administered in the clinic. The formulation, we may go with a high concentration, but because of the limited drug substance, we try to remain in the low concentration, like 50 milligram per ml. We use an IV bolus with the IV infusion. And then as we move to the phase 2, the POC, to subcutaneous. For example, this drug is going to subcutaneous. We still have a dose range from 300 milligram to like 1.5 gram. Again, we had the final clone, and by that time, sorry for the typo, it should be like high concentration. So by that time, we developed the high concentration formulation, like here is 150 milligram per ml. But until that time, the device is still not ready. But we can use, if the low dose, we can use like a 2 ml, like a vial syringe, or we can use an the SC infusion pump. And then as we go along to the phase 3, and that time, the device comes in the picture. We know the dose, and we know what kind of device need to be used. And then we test the device PK study. So this is an example. At each interface of the clinical, there has to be the bridging. We need to understand the PK attributes by the change of the drug substance, the formulations, or the device, and how it's impacting the PK and tolerance. But the trend is now, so I have shown here in the phase 2, we have this subcutaneous, but sometimes that's too late because we need to understand relative BA or other complexities much before. So in the FIH also sometimes we administer the subcutaneous to understand the relative BA between the IV and the SC formulation so that we know the complexity on the PK much before. So this is, again, an example of the roadmap that we use from the phase 1 to launch of the BLA filing. So with that, so I think this is the last one. So thank you, and I think we have time to take in a few questions.
Yes, thank you. Great talk, thanks. Question about hyaluronidase. You mentioned that in rat model, right, when infliximab, it was shown that it increased bioavailability from 50 to 90%, right? And you also mentioned that the increased in clinic by like 20%. Can you please elaborate on an example? And ultimately, what I'm trying to ask is that do you really believe that hyaluronidase does increase bioavailability in the clinic?
Yeah, it's a good question. And I think that's what maybe actually, sorry, I was actually quick, but I can elaborate now. So again, this enzyme is not directly helping to enhance the bioavailability. So this is the hyaluronin, and that is there in the matrix, So that is preventing this high concentration or the high volume, basically the high volume drug when we administer to disperse, right, and to get absorbed. But this technology or this enzyme is helping to disrupt, and so that it creates more channel and more pathway so that the drug is distributed within this hypodermis area and getting absorbed. So basically, indirectly, it is actually helping for this high volume to get distributed and getting absorbed. It's not really helping as a solubilizer, but it's helping in the physiological space to disrupt this enzyme. Is helping in the matrix space to improve or to increase the volume of the drug within this space so that there is less pain and more drug is available for absorption. So this is the way it's helping. So that's what I was trying to get some of the literatures. I know in rats sometimes, in few cases, increase the bioavailability significantly, but based on some of the commercial product, it has shown increase the bioavailability about 20%. So I think in summary, it's more than the PK or bioavailability it's the tolerance, because we are giving a high volume drug and the patient is having more tolerance by using this technology.
Any other question?
So when you do the studies in animals, I can understand that the injection site reactions might be obvious, based on what happens on the skin. But how does the animal respond when there is pain?
Animal is not much responding to the pain, it's more for the local tolerance. Tolerance means the injection site reactions. So we do the mini pig in many cases because mini pigs are more similar to the human. We see any of the injection site reactions. So those are the things. It's not the pain. It's more like like some of the immunological reactions that is happening from the antibodies. So that's the one we see. Yeah, these are more like the morphological areas. So we do the skin pathologies. Okay. Oh. Yeah, it may not be very much correlated with the human because animals are different, so it's difficult. So maybe I can tell you the pain is not, so animals are not a good model for the pain. Basically, human is the best models. And basically, we also do some kind of studies because when you do the PK study, is also the tolerance from the human, all the tolerance and the pain factors and the VA score that we collect to understand the pain also from the device.
Thank you. Any other questions? If not, another round of applause and
Thank you.
we go to the next speaker. Thank you.